A modified agglutination test is used to determine if animal samples are infected with the Toxoplasma gondii parasite. Several reagents and materials are involved in this process: phosphate buffered saline, an alkaline buffer, Toxoplasma whole cell antigen, 2-mercaptoethanol, Evans blue dye, a 96-well U-bottom microtiter plate, a positive control serum that contains a 1 to 200 titer, a negative control serum, and the serum samples will be tested for Toxoplasma gondii infection. A diluted antibody serum sample will need to be created. For mammals, this is a 1 to 25 dilution of 72 microliter PBS and 3 microliter of the sample serum. For birds, it is a 1 to 5 dilution of 48 microliter PBS and 12 microliter of the sample serum. We will use duck samples for this demonstration. 2-mercaptoethanol or beta-mercaptoethanol is commonly used to reduce disulfide bonds and plays an important role in preventing false positives. The antibody we are testing for is Toxoplasma-specific IgG. Non-specific IgM antibodies can interfere with the MAT test and produce false positive results. 2-mercaptoethanol is used to cleave the disulfide bond present in IgM, thus destroying the IgM antibody and creating specificity for the IgG antibody. This is an example plate for the MAT test. Samples will be placed in the first 10 columns of row 1, with the controls added to the final 2 columns. A serial dilution of 25 microliters at a time will be performed to reach a 1 to 640 dilution. The excess 25 microliters will be discarded. Begin by first properly labeling all microcentrifuge tubes with the sample number and by labeling the U-bottom microtiter plate. After allowing the samples to properly thaw, vortex them for several seconds to prevent settling. Add 48 microliters of PBS to each microcentrifuge tube. Add 12 microliters of each sample to their appropriate microcentrifuge tube, including the positive and negative controls, and mix by pipetting the solution several times. Vortex each microcentrifuge tube for several seconds to ensure even distribution in the PBS. Place each tube into the microcentrifuge and allow to spin for 30 seconds to ensure all of the solution is in the bottom of the tube. Add 50 microliters of each diluted sample to their respective well in row A of the microtiter plate. Add 25 microliters of PBS to the rest of the wells using a multi-channel pipette. Perform a serial dilution using the multi-channel pipette. Transfer 25 microliter of solution from each well to the next. Discard the excess 25 microliters of solution after the final dilution. Next, we need to prepare the antigen mixture in a new 10 milliliter centrifuge tube. Add 2.5 milliliters of the alkaline buffer. In a fume hood, add 35 microliters of the 2-mercaptoethanol to the centrifuge tube. Mix well by pipetting several times. Add 50 microliters of Evans blue dye to the centrifuge tube. Mix well by once again pipetting several times. Gently shake the antigen to ensure even distribution within the solution. Add 150 microliters of the antigen to the 10 milliliter centrifuge tube and mix well by pipetting several times. Add 25 microliters of the antigen mixture to every well on the plate. Make sure not to touch the sample in the well while doing so, or you will need to replace your pipette tip. Carefully seal the microtiter plate using tape. 2 pieces of tape should be enough, and you should crimp the edges to allow for easier removal. In a container, place one wet paper towel. Place the taped microtiter plate on top of this paper towel, and then cover the plate with a second wet paper towel. Seal the container with Saran wrap. Ensure that the container is properly sealed. Store the container in an incubator set at 37 degrees Celsius for 16 to 24 hours. Remove the microtiter plate from the container and gently remove the tape. Carefully dab the plate with a paper towel to remove excess water. Place the plate above a mirror with light shining through so that you can clearly see the bottom of the wells. A pellet indicates a negative result, while a lawn indicates a positive result for the Toxoplasma gondii antibodies.